Here we are on Chapter 7. We're doing cellular respiration, and we have looked at already a couple of things. So let's take it from there. Let's see if I can move this slide forward. That would be nice. There we go. So remember that we are really talking about how we, and looking at it from the individual cell standpoint, how we get energy. And so we see here that we know that we're looking at metabolism. Metabolism involves taking in certain materials, breaking them down, and then restructuring them into something that's useful for the cell. So in the case of this picture here that you see a nutrient that's coming in, and then it's going to be taken into the cell, and then in being taken in, we're going to extract some useful materials and some energy from it. Some of those materials may end up as building blocks of some anabolic pathway, and then when we're looking at the breakdown, we're looking at catabolic types of reactions. So we're really focused specifically, though, on glucose. So we're looking at glucose metabolism and the production of ATP. So here you can see, as you remember, that glucose first has to get into the cell. It's going to be able to get in. There you can see insulin actually has a role in all of this, but glucose will enter the cell through the plasma membrane, through those transporters, the GLUT4 or GLUT transporters that help to bring it in. So that's just a quick review. And then we went into this, which looked at what happens just in the cytoplasm or cytosol, where glucose is converted through a series of 10 enzymatic steps. First in this preparatory phase where glucose, we actually use ATP, so the cell uses ATP twice. And so these kinases that you see here are indicating that ATP is being used, that this is a coupled reaction, taking us from glucose to glucose 6-phosphate. And then we rearrange glucose 6-phosphate into its isomer using this enzyme, the isomerase, which creates fructose 6-phosphate. We have another conversion here, again, that coupled reaction using ATP, where the coupled reaction releases enough energy to push this reaction forward. Also, though, the phosphate is used as a part of a substrate onto fructose 6-phosphate. Now we have fructose 1,6-bisphosphate. And then ultimately we get two molecules that are almost symmetrical, and then we have an isomerase again that rearranges them so we have that symmetry. We end up with glyceraldehyde 3-phosphate, or what we're calling G3P. So far, so good, I hope. Here we go, and now we're going on to what we call the energy payoff phase. And so we're extracting energy from that G3P. So this was that product at the end of step 5. Immediately, the energy that's extracted is the electrons that are taken away, and you can see that takes the oxidized version of this carrier, NAD+, and it converts it into the reduced version. So now we have our NADH, our first NADH. We go ahead and now in two other steps. So step 6, we get NADH. Step 7, a kinase is involved, but in this case, we're using that kinase to put a phosphate onto ADP. And so we're getting an ATP. And then down the road here on step 10, another kinase is doing the same thing. So we get an NADH, we get an ATP, we get another ATP, but remember that happens twice because at the end of step 5, we ended up with 2 3-carbon glyceraldehyde 3-phosphates, or G3Ps. So you can see those, there's the 3 carbons. All right, moving on. So now we are going into the link reaction. So we know that one of the products of, actually that's bad because I didn't tell you that here's our pyruvate. So I just want to point that out. So pyruvate is a product at the end of these 10 steps. You see it's a 3-carbon compound. It has lots of oxygens on it. Now we're going to go forward, and we see the pyruvate again, and the pyruvate is going to get transported in to the mitochondrion. So immediately what happens in this reaction that you see here kind of spread out is that we're going to lose a carbon dioxide. And if you look over here, that's been highlighted. We're going to generate another NADH. Okay, so this is an oxidation reduction reaction, taking the electrons away from that original pyruvate and now creating this thing called NADH. And that is all in combination with an enzyme called coenzyme A, which yields at the end this molecule called acetyl-CoA or acetyl-CoA. We can call it either one. Where all this happens, just to understand that when we get into the mitochondrion, or mitochondria, which is plural, what we see here is its structure, where we have an outer membrane. We have an inner membrane that's folded within the mitochondrion. Those folds, back and forth folds, are actually called cristae. We have inside, between the two membranes, we have something called the intermembrane space. And then within sort of more the central area, and here we see this just blue, we have what's called the matrix. So the other thing you're seeing in here, which we'll get to later, is these things called ATP synthase, which if you see the ASE, you're saying, whoa, there's an enzyme. So we'll look at that soon. All right, so next. So we're going to go to here. So here's the acetyl-CoA that was generated in the link reaction. You can kind of recognize here it has that sulfur on it. Remember, there was an SH originally on coenzyme A before it formed into this. So it was pyruvate. We made then acetyl-CoA. Acetyl-CoA is missing one of its carbons. So pyruvate is missing a carbon, because remember it was 3 carbons to start with, now we have 2. And so just to remind you, we're doing some counting now of where carbons are going. So acetyl-CoA has these 2 carbons, and it's going to enter into this cycle that's called, actually 3 different names, but the one we'll probably refer to it mostly as is the Krebs cycle. This is taking place in the mitochondrial matrix, that part we just looked at. So when acetyl-CoA comes in to the cycle, what you see here is there's a reaction that happens right here with water, some hydrolysis. But more than that, what it's doing is it's joining this molecule right here. This molecule is called oxaloacetate. It has 4 carbons. Acetyl-CoA has 2 carbons. And when that joins, what we get is a molecule called citrate. So citrate is what we're seeing right above here. Here's citrate, if I can get my cursor to work. There we go. So here's citrate. And that coenzyme A has actually been removed now. So now we've got citrate, and this will go up and get recycled and used again. But here's our citrate. 6 carbons. 6-carbon citrate is going to get converted into another compound, but the conversion is just a rearrangement. So what's the enzyme? It's just rearranging. And then between this citrate and this step, we had that rearrangement, and now we're going to extract some electrons. So here's NAD+. It's going to remove some electrons, forming NADH, right, the reduced form. And in that process, we're going to lose a carbon dioxide. Now, you do not need to know most of these terms. The only names that I really would like you to know is this first one, oxaloacetate, and obviously acetyl-CoA, but the forming of citrate. So oxaloacetate joins with acetyl-CoA, forms citrate. We rearrange it, We get down to this next step, we take off some electrons, we lose a carbon dioxide. Right away, next step happens again. Same thing here. We're going to go another step. So now we've generated 2 NADHs. We've lost 2 carbon dioxides. And now we're down here at the bottom. Notice what's happening here is we get this product of that reaction is going to move this direction. And you don't have to worry about anything else on here, except now we're going to get an ATP out of it. Now you may say to yourself, that's not an ATP, and you're right, because there's a different form that ATP can also be found in certain cells, and that's something called GTP. So ATP and ADP, GTP, and GDP are for our purposes equivalent. So here we're forming a GTP, and that GTP can later be converted to ATP, but that doesn't matter right now. So now here we have an ATP or a GTP that's formed. Here's the first time we see FAD. Remember, FAD's also an energy carrier. It's part of the redox reactions. And this next step, what we see is we generate an FADH2. We go a little further, we go a little further, we get another NADH. And now, at this point, when we're already back up here, now we've regenerated oxaloacetate. It's ready to pick up that next acetyl-CoA from the link reaction and go through this whole cycle again. Now, just like when we looked at the second part of glycolysis, when you're considering a single glucose molecule going through this whole set of steps, remember that now we have 2 pyruvates that go in and they form 2 acetyl-CoAs. And so this process is going to happen 2 times for every 1 glucose that comes in. And that's the Krebs cycle.